All right. Yep. So today we have John Fowler, who was kind enough to hop on and sort of offer his presentation last minute. Sorry, I'm just rearranging things here on my screen. Uh, yeah, so he's going to be talking about maize pollen today, and his presentation is going to be about the successful predictive modeling of pollen fitness phenotypes. Uh, oh,
Oops, did I, let's see.
oh, no, sorry, um, my computer's being weird.
Should I try the share now?
Yeah, go ahead and share. Yep,
Okay.
sorry. Tried to read the whole title, and yeah,
Okay, well, I've got it on the first slide here.
yeah.
Uh, anyway. Okay. All right. Let's see if I can get this to work. All right, can folks see that okay? Um, let's see, continue to share. And all right, so yeah, so thanks to John for letting me uh, sort of do this very last minute um, and talk about maize pollen, which I will talk about for hours, I guess. So what we're trying to think about in my lab is really, I think of our mission as being, how does this plant work? Here are these maize pollen grains. They've been put into liquid media, and very rapidly they begin to grow their pollen tubes, which are really lovely and amazing. And one of the, I mean, you can see these by eye growing. They're one of the fastest growing plant cells known. Um, but the interesting thing about pollen is that there's this subtitle that I'm sort of using in almost all my talks, and that is a reminder that we tend to think of them as very short-lived, very ephemeral. Of course, they're critical for reproduction, but you can really think of them as independent plants as well. And I think that some of the work that we're doing is sort of highlighting this. Um, Before I get too far into this, I want to mention the key contributors here. So Zuzana Velipkova is a longtime senior FRA in the lab, and she's coordinated the genetics. And then a lot of what I'm going to talk about is work that's collaboratory work with Molly McGraw's lab. And Sebastian is the graduate student who is actually doing, as you know, most of the work. And both of them came to the Maze meeting last year, and Sebastian presented an earlier version of this in the development pre-meeting. So if you were there, you've heard part of it, but has since elaborated on that. And I'll mention Cedar, Marilyn, Sam, and Diana real briefly because they did work that also contributed. Also, the other reason to sort of fit this in is that we're sort of in the final stages of preparing this into a manuscript. So any feedback that you can provide, either questions afterwards or even by Slack, I'm on the Slack now. I'm very happy to have that because it'll help us finalize that manuscript. Okay, so here's the thing to remind you to start off with. I teach intro biology, and all of the med school students are just unhappy when I bring this up, but it's really critical, and that is we talk about plant reproduction and this alternation of generations. And that is that plants have this gametophyte phase, which is haploid, and of course, the big difference between animals and plants when thinking about this haploid phase is that for plants, you have uh, development, you have gene expression, you have response to environment. And what that means is that the haploid genotype actually can make a big difference in terms of well, selection and phenotypes. And so that is going to show up quite a bit in it's really sort of the central point of how we approached this work, is recalling that pollen, as the male gametophyte, is haploid. But of course, even though I'm sort of calling it this independent organism, this independent plant. It's a relatively simple organism, and it has a very focused mission, right? It does not do photosynthesis. It doesn't have to harvest nutrients from the soil. That comes from the parent. And it has this single mission of being the truck, right, that carries the sperm cells from one plant to the other. Um, there are three cells in the maize pollen grain, the vegetative cell and the two sperm cells. And the pollen tube itself is, of course, the product of that vegetative cell. Um, in maize, of course, it's highly competitive, lives in a highly competitive environment, and that the estimates that I've seen are that a single maize plant can actually produce a couple million pollen grains, and a single silk, there are several hundred that can be captured on that silk. And so they can generate pollen tubes, and then they can compete with each other. And of course, the final part of that is to bring those sperm cells down to the embryo sac for double fertilization. So again, maize pollen is pretty extreme in its sort of both how rapidly it grows, how far it has to grow through the silk to reach the embryo sac, and in how many pollen grains it produces. And again, it's haploid. So in terms of thinking about pollen and how it works, since I'm a geneticist, I'm interested in phenotypes. But for the phenotype, you want to have a way to actually measure it. So one of the questions is how to measure pollen phenotype. And of course, you can plate pollen grains and measure how quickly they grow their pollen tubes. But uh, it's a lot easier to approach it from, again, a genetic standpoint. And I'll remind you that, well, Mendel is awesome and has this really very simple uh, uh, prediction about progeny ratios. And that is if you have a heterozygous plant with a dominant and recessive allele and outcross it to a homozygous recessive, then because of segregation, you get one-to-one segregation of the progeny that you can see really easily on this ear. So, and the numbers are really high for a maize ear. So you can clearly see this type of, segregation happening all the time. However, you can take the same cross, and sometimes you can see an effect like this. And that is you have a heterozygous parent uh, and make the outcross, but in the progeny, you see a much reduced transmission of this dominant C1. And you can figure this out just by counting the anthocyanin kernels on the ear. And so there's you can determine this is not due to epistasis or things along those lines. Rather, it really is a reduced transmission of that purple anthocyanin C allele. So how can that happen? Well, there's a variety of ways, but the way that I'm going to be focusing on is the following, and that is if you have a mutation in a gene that affects pollen that reduces its fitness, then it doesn't compete as well. And so then you have these pollen grains are going to be actually less competitive. They'll do their job at a lower frequency. And so you end up with a difference from one-to-one segregation. So these deviations from one-to-one are explained by that mutant effect on the haploid gametophyte. And furthermore, this transmission rate is actually, you can think of as a quantitative measure to get some sense of the severity of the phenotype. So it's a quantitative phenotype for this particular mutation. The problem is that this only works easily if your pollen mutation is linked to a nice endosperm marker. Otherwise, it's really difficult to count. You can do it by PCR. And that's where this great maize resource comes in. And this is the DSGFP mutant population, which was generated by Hugo Dünner and Charles Dew, where they took this engineered DSGFP, which has a element which has GFP on an alpha Zein promoter and makes these really amazing, very easy to see with the right sort of lights, fluorescent kernels. And so because they have these distributed throughout the genome and mapped to different genes, you can actually use this as a way to assess easily the transmission of any line, and perhaps lines where you have a single gene insertion that disrupts that gene. And so the DSGFP element is not only the mutagen, but it also is the marker that allows you to do the counts that give you the transmission rate. So then this is a super simple assay in maize because it's so easy to make these crosses. And that is, you have a heterozygous pollen parent. It's going to produce half of the pollen are going to be wild type, so not green. Half of the pollen are going to have this insertion, this DSGFP, where the gene itself is mutated. It starts out 1-to-1, you do the pollination, competition occurs, and then you just count the GFP kernels on this ear to calculate the transmission rate that is associated with that specific mutation and gives you an idea of its effect on fitness. So this is, like I said, it's relatively easy to do. And one of the issues is that you have to be able to quantitate it. And so this is, I just have to shout out to Cedar Warman, who's a former PhD student in the lab who invented this maze scanner, which totally changed the way we do the work in the lab, where you can basically image a spinning maze ear with a movie and then make this projection of the entire surface of the ear, which really facilitates making the counts that I'm interested in. That is the GFP versus the purple wild type. And also Cedar, and then follow up by Marilyn and Diana, made a automated phenotyping pipeline called Ear Vision, which allows that counting of these images to be done in an automated way. And that means that we can generate this type of data where you have a particular mutation in this yellow stripe transporter. And here is the cross with the pollen parent is the heterozygote. Here is the cross for the same mutation with the ear parent as the heterozygote. So here Mendel's prediction works. Here it gives an idea that this particular gene is really important for pollen. And here's a different mutation, BAG1, which Diana will actually talk about in a few weeks here at the seminar series. Again, the female cross through the ear is 1-to-1, and the pollen cross is reduced in the transmission rate. So we can associate these numbers, these quantitative phenotypes, with specific mutations and specific genes. So, and we can do this at a scale that's, it's not like a RNA-seq, but it's fairly large. And so the rest of this talk is basically what you can do with a lot of that sort of phenotypic data that relates the genes to quantitative phenotypes. So when thinking about approaching this, one of the things as a geneticist that's sort of been annoying over the years is that I'd write to the co-op and request a mu insertion into a gene that looked really interesting, and then it would not have any sort of pollen phenotype. And so, the problem is we have now all these data associated with genotype. Here's the 26 NAM genomes that have been sequenced. Here's this really nice paper from Justin Wally and Steve Briggs's lab, where he looked at RNA and proteomic measures across 23 different tissues. And even though we have all these omic scale data, our ability to precisely predict the phenotype from these is still not great. I mean, we can make some good guesses, but it'd be really nice to have like an equation, like a formula that would tell us what are the most likely genes to actually have phenotypes when mutated. So when we think about predicting phenotypes from data, we can think of it in two different ways. One would be mechanistic, where we understand all of the molecules that these genes make and how they interact. And then from that, we can actually make some predictions about some phenotype. The other way to do this is more of a systems approach, and that's what I'm going to be talking about. And this is sort of looking for overall trends that are sort of high level, and then looking for correlations that give us some ability to make predictions. And the reason why we thought that this might be possible with maize pollen is that in Cedar's original paper, where we talked about ear vision and thought about how it could be useful, we had a pilot experiment where we looked at the transmission rate phenotype of 32 different pollen genes. And what Cedar found was that as you mutated genes that were more highly expressed based on RNA-seq in the pollen, that the transmission rate for mutations of those genes actually decreased. And this sort of made some sense in that if you have a gene that's expressed really highly in pollen, well, maybe that's an indication that it's really important in pollen. And so when you take it away, you're actually going to make a more severe phenotype. So this was a pilot, and it was a select set of genes. And so there were several questions here. One is, does this correlation hold for a larger data set? And secondly, are there other genome scale measures that could sort of combine with expression data to help predict genes that have fitness effects? Okay, so this is where Molly and Sebastian come in because Molly is computational biologist, expert in employing machine learning approaches, and Sebastian is in the bioengineering PhD program here at OSU and in Molly's lab, and was interested to try some machine learning approaches on this. And the other part of this talk is just to highlight how awesome, of course, Maze GDB is, and also sort of the nice data sets that Maze people produce and we've actually used in our modeling have turned out to be really useful. Okay, so I'm going to spend the rest of the talk talking about this machine learning project. And so this is all coached by Molly and Sebastian. So they're here on the seminar. And so if you have any, they'll sort of help fix me if I say something wrong and answer questions at the end um, if you have any on the details. So just a brief idea about how machine learning approaches can work. And that is that with a machine learning approach, you take a variety of quantitative measures. These are called features that can relate to a variety of things: gene length, mRNA level, amino acid composition. Then these go through the machine learning process using a model that's logistic regression, where all these features are placed with weights that then generate a score that helps predict whether the outcome is in one class or not. So this is going to be classifier modeling, where we have two different possibilities. One is we have a gene that when mutated will have a high fitness effect, and so reduced transmission rate. And the other will be just a regular Mendelian transmission rate or no fitness effect. And the nice thing about a machine learning approach is that as long as you have a training data set, one that has some example data to actually generate this, it can search the various features that are out there, such as all these omic scale types of data, to generate a model that has some success in this. Okay, so this successful model can both integrate a wide range of data types, so like the three shown in here, and it can also provide insight into the underlying drivers for what is sort of associated with those differences with the phenotype in this case, and that is that which features are most informative. So this is, again, not a mechanistic talk. Rather, it's sort of gonna hopefully give us in the end, those some features that are really interesting that are worth further exploration in follow-up experiments. Okay, so what about this training data set? So we use this DSGFP to assess the transmission rate for a large number of pollen expressed gene models. So we targeted a little over 400 genes, which are mapped to as insertions into pollen expressed gene models. And when I say pollen expressed, these are all genes that are expressed above background, either by RNA measures or protein measures. This Wally paper had proteomic profiling for pollen there, which is really useful. We went after insertions in the coding sequence because the idea was that those would be the ones that would most likely have the most severe effect, be knockouts. And I have to say that there is a bit of a limitation in this data set that's inherent to the way that the DSGFP population was produced. And that is the original mutagenesis was via a pollen cross. And so just unfortunately, that means that in that original mutagenic cross, they selected against insertions that perhaps would be most interested in, that is insertions that cause the largest effect. However, as you'll see, we definitely found some genes that were very informative in this, but it's worth having in mind that limitation. So the data were gathered over 7 field seasons. At this point, nearly 5,000 ears harvested in imaging, and 1.5 million kernels phenotyped. And we don't have, obviously, second alleles for every one of our insertions, but for the genes where we have 2 or more alleles in the same gene, then what we've seen is that you get similar quantitative phenotypes. So at least to the extent that we can, we have some genetic validation for these data. Okay, so in this one, this one plot shows a lot of work, summarizes a lot of work, all that crossing, statistical help from Duo, Luis, and Michelle in the statistics department here. And so each of the plotted points on this graph are a separate allele that we have phenotyped via our ear vision. And it gives the transmission rate. Here's 1-to-1 here at 0.5. And then what you'll see is that you do get sort of a tail that it tends this way. These are all genes, these are all alleles with reduced transmission. All of these have been PCR validated, and the estimates of the transmission rate are via generalized linear model. So we've got a total of 309 validated insertion alleles. Notably, none of them are 0% pollen transmission, which sort of makes sense given the generation of that population. But we do have a few that have pretty strong effects. And I think it's important to note here that this measure of fitness, so these are all the genes that are going to have reduced fitness, captures a range of cellular phenotypes, right? Some affect sperm cells, some affect pollen tube. Anyway, there's a variety of things, and all we're getting with this is this outcome, right, of what's the quantitative effect on fitness. The other thing I'll note here is that we made a call early on to leave out in the modeling these alleles that are represented in blue between 45 and 48% because they didn't have a really strong fitness effect, but we couldn't really say that they were Mendelian either. So only the modeling only works with the green and the red as a fitness effect and no fitness effect alleles. Okay, so as I mentioned, here's this training data set that we're going to use for machine learning. We've got no fitness effect, which has Mendelian, and then a fitness effect, which is 45% or less GFP. For the modeling, when we narrowed it down, left out those blues, left out the duplicates, we had a total of 227 unique gene models, and 10% of those which have these high fitness effects. And Molly and Sebastian are not shy to tell me that this is a really small data set for machine learning. But it was a lot of work. And so, you take what you can get. And Sebastian then used this to explore a range of features to see which could be predictive for this phenotype. And it turns out that expression data is the most informative, and I'll be talking mostly about that as I go through the rest. And it's important to note here that the proteomic profiling data also are a limitation because for whatever reason, the profiling only captured a subset of the genes. So we only have measurement data for, again, a subset of the 227. Okay, so before I go any farther, I need to talk a little bit about metrics for determining success with the machine learning models. And for that, the primary metric being used is the area under the receiver operator curve, or AUROC. And the main thing to know here is that what this is doing is trying to balance sensitivity, that is getting a high true positive rate without getting too many false positives. So also trying to take into account specificity. So you plot this out, area under the curve, and then the sort of rule of thumb here tells you 50% area under the curve is basically no information from the model. 100% is going to be your perfect model, and anything in the 80-90% range is going to be a well-performing model that you're pretty excited about. So pretty early on, Sebastian's work showed that we could actually get quite good models that were successful in predicting whether a particular gene in this training set was would confer a transmission rate defect or not. In the end, these are the three best models that he's generated. One is a genome model, one is expression model, and one is a combination model that includes data from sort of genome only. So this has no expression data in it. It's only features that are derived from genome sequence. Expression model is only based on RNA and protein expression data, and then the combination model brings in data from both sets. You'll see a few of these types of graphs over the rest of the talk, where this gives you this average AUROC for a particular modeling run. This is done with threefold cross-validation, where you take your data set and split it into thirds and then use each 2/3 as a way to model and then validate with the other third, and then go through each of those partitions. And the box and whisper plots give you an idea of sort of how stable the modeling is, and that this is random seeds for determining how those partitions are generated. So there's 1,000 modeling runs for each one. And what you'll see is that the combination model is the best, the expression model is next best, really strong, and the genome model is pretty good. So I'm going to talk a little bit about how we got to this. And one of the early questions we had when Sebastian was having some success was whether the RNA data or the proteomic profiling data were more informative in terms of generating a successful model. And I thought for sure it would be the proteomic profiling data. And it turns out that this Wally paper sets things up really nicely for actually asking this question, in that they surveyed 23 different developmental stages, one of which is mature pollen, and these are the other ones that were covered in the paper. And for each stage, they have paired samples. So there's an RNA-seq sample and there's a proteomic sample. And then there are three summary features. One is this max measurement, which is the maximum of all across all 23 tissues for the RNA-seq, or the maximum for the proteomic profiling. One is average across all these. And then breadth is the number of stages that have some sort of signal. And when you model restricting yourself to just these features, it turns out that the RNA-only model does a little better than the protein-only model. However, this 3-point difference is not huge, not significant, and also you'll notice that these aren't really the best models. These are in the 70s for the AUROC. And the other thing that was sort of confusing or less good, I guess, about this modeling is that when you put the two data types together, you really don't see any improvement. And this was strange because the idea is that the machine learning process would actually find ways to combine all these different RNA and protein data to give you the best model. And so this led us to look a little more deeply into the actual sort of guts of the models. And to talk about that, I'm going to, again, actually go into a little bit more about how the modeling works, and particularly the feature weights. So in the final model, what you end up with is particular features that are very informative that are selected as being predictive of this difference. And each of those features is accompanied by a beta coefficient, which is the weight. And this is going to have a sign, that is, it's going to be either positive or negative. A positive weight is going to weigh it more towards fitness effect. A negative weight is going to weigh it more towards a no fitness effect. And then the magnitude of that is going to also be optimized during the modeling process. So again, we can sort of think of this as a bit of a balance in that if you have a large weight, that's a positive weight, that'll multiply with whatever feature this is, that will then sort of outweigh any negative, and that will point toward a gene that has a fitness effect when mutated. On the other hand, if your feature that's associated with that large weight is really low quantity, then it's not going to come to much weight and won't balance it toward fitness effect, but rather, in this case, maybe the negative weights will point towards a particular gene having no fitness effect. So one of the things that Sebastian noticed as was going through this early modeling, looking at RNA and looking at protein, is that there was a pattern that showed up that was really striking. And this is the clearest example of that. And this was with modeling with just the proteomic data. And that is, if you, in these models, you had a few other features, but you always had protein expression in mature pollen as a highly weighted positive weight. And then you had another feature that had a negative sign. There was maximum expression in the protein across all tissues. Okay. And so if you think about this, and the other thing that we noticed was that these two were opposite signs, but similar in magnitude. And so if you think about this, what it means is that when you have a maximum expression that is not pollen, and so you're going to have maybe some expression in pollen, but somewhere else there's more expression, that's always going to give you no fitness effect. If mature pollen is the maximum, then these are sort of going to cancel out, and then the other features can weight it toward fitness effect. So based on this, this sort of says, again, if it's expressed someplace not in pollen, then it's probably no fitness effect, which seems strange that expression in, root or something would inform something about pollen phenotype. So we came up with this hypothesis is that what the model was sort of trying to get at was the opposite, and that is a way to calculate expression specificity. And that is maybe what would be the most effective way to sort of inform this model is actually come up with a measure of specificity of expression in pollen. And this would be a positive weight feature over on this side that would really help predict pollen fitness genes. And so to test this hypothesis, we engineered 3 different additional variables looking at sort of a larger second level for expression, and asked the question, do these features boost model performance? And if they do, then that would support the idea that specificity is really important. So the first feature I'm gonna call PMP, and basically this is a binary pollen max protein abundance. If the tick's tissue of maximum protein expression is pollen, it's going to get a 1, otherwise it gets a 0. A similar sort of approach is to, or trying to capture a similar type of uh, concept is this pollen protein abundance ratio, where you take the ratio of pollen protein expression and you ratio it to the maximum non-pollen protein expression. And this is going to give you an idea of how specific in a quantitative measure its pollen expression is. And the final one is called non-pollen specificity ratio. And this is trying to capture specificity in something that's not pollen. So you take the largest ratio of non-pollen expression and you ratio it to the next largest, with the idea that this would be potentially a negative, have a negative feature weight. So this is a rather complicated uh, slide, but it basically supports the idea that these specificity measures are major boosters to our modeling success. So on the left here, this is the AUROC for models that didn't have any specificity features, but were really the best with the expression data that we had. And these two models, which increase by 5, 6 points, are when you add in that binary, the PMP, or the ratio that's trying to capture pollen specificity. And when you look at the feature weights, and here's the PMP with that binary, and you can see, again, it's a positive feature weight. It's a pretty strong one. And when you get to the ratio, which captures pollen specificity, if you log transform it, it becomes a really strong positive predictor. Um, now, the non-pollen specificity wasn't really helpful on its own, but it turns out that in the end, the expression model, which is the best model that Sebastian came up with using only expression data, turns out to have 3 features. The first is this log of the pollen expression, the protein abundance ratio. As a negative feature, the maximum RNA expression value across all the data is a strong negative weight. And then the other negative is this non-pollen specificity ratio. And these 3 features, if you use them in the model, give you an AUROC of approximately 0.9. So very useful in terms of modeling. And this plot, which shows that log of the pollen expression ratio on the x-axis relative to the estimated transmission rate for each of our different alleles, shows why that is in a really graphical sense. And that is, you can separate the genes that we assessed into basically 3 categories: a pollen-specific category, a non-pollen specific category around 0, where you get expression both in pollen and some other tissue, and then a pollen minimal. And what you can see is that most of these pollen-specific, most of our transmission rate mutants, which are in red, are in this pollen-specific category, and it has relatively few that are Mendelian, whereas the pollen non-specific and the pollen minimal have lots of the Mendelian and very few that affect transmission rates. Okay, I am not keeping track of the time, so can somebody tell me how far along we are?
We've got about maybe another 10 or 15 minutes that we can follow.
Okay, all right. So, so again, this argued that the specificity was really important and really useful in defining which genes, predicting which genes would have pollen phenotypes. So to get to the genome and combination models, Sebastian was faced with the problem that these genome-only features, there's quite a number of them in Maze GDB. And you can separate them. Here are all the types of these feature sets that are available. I've sort of grouped them into DNA ones, protein ones, and ones that are related to variation in evolution, like synteny. These are sort of amino acid composition types of descriptors, and then dimer, trimer, gene structure, DNA methylation. So these are too many features to throw into the modeling all at once. So Sebastian took an iterative modeling process where he generated models for each of these using the multiple features in each of these subsets, and then categorized each subset as either being poor, moderate, or strong in terms of its predictive ability, and then went through the strong, so that would be the green, and the moderate, that would be the yellow, and looked for specific features within those subsets that contributed substantially to predictive success, and then pulled those out and did a final round of modeling, sort of brute force, multiple sort of tests with a streamlined set that actually found the most informative features. And these then went into the genome-only model, so again, based only on genome sequence, or the combination model, where you took the expression data and put it with data from the genome. And this is how, this is just going back to this graph, comparing these three different models and showing the features that ended up being used in each of these models. And I don't have time to go into some of the detail about these, and the genome and combination. Some of these features are less easy to interpret than the expression data. The CTDD is a protein amino acid hydrophilic, sort of describing the hydrophilic distribution of hydrophilic acids in proteins. But again, this combination model was really interesting because it gave the best AUROC, and it combined that log of the specificity value with a couple of interesting evolutionary variant type of features. Notably here, these are, if a gene is a singleton that is not paired in the maize genome, it is more likely to actually lead to a phenotype, which makes a lot of sense. Okay, so the final question we wanted to get to is, how well do these models generalize across the maize genome? It turns out that then you can just take these models and apply them to the rest of the genes in the genome, and then sort of, in this case, we plotted them relative to this really useful pollen specificity ratio on the x-axis. What you can see for the genome model is when you go out past our training set, it doesn't look really attractive because it actually predicts a lot of fitness effect genes in genes that are barely expressed in pollen at all. As expected, the expression model, of course, picks up a bunch of genes that are highly pollen specific, but not all of them. So that's sort of intriguing. And the combination model really looks perhaps the most attractive in that it does give you a lot of these pollen expressed specific genes, but it also gives you some in the pollen non-specific genes. And when you look at across the genome how these data sets intersect, you can see that they are picking up basically three different groups of genes. So this is intriguing, and it sort of, again, makes us think that maybe these two generalize relatively well. But we wanted to look for a way to really test that more quantitatively. So remember that, I mean, the whole sort of point here is that pollen mutations don't transmit well, they get lost. And so a prediction would be heritable mutations in these types of genes should be more difficult to recover in populations, in maize populations. And so there are several maize populations out there where we actually know exactly where specific transposable element insertions are located. And so this prediction is that if we have pollen fitness genes, they should be associated with reduced frequencies of transposable element insertions in these sequence indexed populations. And so we went to the Bond mu population, which these are data published by Caroline Micron associated with the Hochholdinger group, had a paper in 2020, and then a recent one in 2024 that really greatly expanded this resource. So one awesome thing about this population is that it was generated with the mu active as the pollen parent for the entire resource. And so again, if it's pollen fitness genes, these should be present at reduced frequencies in that entire resource. Their resource has over 425,000 insertions that are mapped. And furthermore, in this last paper, they did this really nice job of mapping specific insertions relative to particular partitions for the specific gene models that they intersected. So are they in the exon? Are they in the intron, the 5' UTR? And what allowed us to do is Sebastian generated what we call the bond mu test, which asks the question, if you look at genes that are predicted to be pollen fitness genes and then compare them to the no fitness genes, is there a reduced frequency for insertions in each of these particular partitions? Okay, with a prediction that we should see fewer. These are the control. So this is the training data set, and you see exactly the trend that you would expect in that the fitness genes, these are, of course, ones where we know that there's a fitness effect based on the DSGFP data, have reduced insertion rates in these promoter core, that's the first 100 base pairs, 5' UTR, the exon, intron, 3' UTR, with the largest effect, most significant effect, the exon, which makes total sense in terms of its ability to interrupt the gene. So then we use the same test with the genes that were predicted in the other parts of the genome based on the models. Okay. So the genome model, again, did not do that well. Most of in terms of expanding across the genome, there was not a lot of difference when looking at the bond mu population. However, both the expression model and the combination model showed the trend we predicted. And that is that in genes that the models predict are pollen fitness genes, you see fewer insertions, a frequency that's lower in those genes. The statistics here come from a 1,000-fold permutation test where Sebastian basically randomly shuffled around the fitness, no fitness labels for these models, and argue quite strongly, we think, that the models are actually, these two models are generalizing quite well across the genome to pick up genes that are likely to have fitness effects. It's important to note that there's one other possible explanation for this, and that is that what we're picking up here is not reduced insertion due to a effect on pollen fitness, but rather reduced insertion rates because these genes are not sort of available. They're not, maybe they're in heterochromatin, and so they're not being inserted in for more of an epigenetic reason. But that doesn't make a lot of sense if you look at the exon and intron here for this combination model, because one would predict that those would have similar effects if it's sort of an availability effect, then one would expect it to affect both introns and extrons equally, whereas the combination model gives you this really strong effect that's really specific for exons. Okay, so just to finalize and summarize, basically what we think is, given an appropriate data set, even a relatively small one, that using a machine learning approach can successfully help predict mutant phenotypes. We think that two of our models, the expression and combination models, are actually generalized well across the genome. And notably, the combination model takes the expression data and combines it with data which is complementary to that we can get from the genome to get to that highest AUROC. And it also gave, I think, the best results. Looked to me like the nicest pattern in the bond mu test. Measures of both gene expression from RNA-seq and proteomic profiling can both contribute. And this expression specificity, most notably the protein ratio, was really critical for generating the best forming models. And so we'd argue that this is a really key marker for genes contributing to pollen fitness in maize. And I'll go to this figure, this plot again, because one of the points to note here is that these two sets of genes, the pollen specific and this pollen non-specific genes, have really similar levels of expression. So it's not that the pollen specific genes are more highly expressed. They're just more specific, and this totally makes sense that this would be a marker for a gene that's important for pollen. But the thing that confuses me, and still I'm a little confused about, is why there's so many pollen non-specific genes that when you mutate them, they don't seem to have much of an effect on transmission rates. So I'll finish with a couple speculations. And the first is the only sort of answer, or sort of, I don't know if it's a favored answer, but it's one possibility for why you get all these non-specific genes that are expressed in pollen that are going to have an energetic cost. Maybe it's difficult for genes that are expressed really broadly to get a specific sort of regulatory mutation so that they get turned off only in pollen. I'm really not sure there. But the other thing is that there's an actual evolutionary implication to these data, and that is there's a longstanding hypothesis that the haploid phase, particularly pollen, actually helps acts as sort of a genetic filter and helps purge deleterious mutations that might affect the sporophyte, because since it's haploid, if you have a gene that's expressed both in the sporophyte and the male gametophyte, then if you have a deleterious mutation, that pollen is less fit, and then it doesn't transmit. And so then you sort of have this purging that happens due to just the presence of having that gametophytic phase. However, our data argue that at least in maize, that effect may be somewhat limited, in that the genes that are really contributing to pollen fitness are the pollen specific genes, and the non-specific genes are not really influencing pollen fitness. And so these deleterious mutations would not be screened out. Okay, so I hope I made it in time, and I'm happy to take any questions.
Awesome. Yeah, it was a really neat talk. I think Jeff actually kind of started us off with the
Mm,
questions. If you want to hop
Hey,
on.
John. Yeah, that's super cool. And the phenotyping platform is really amazing. Um, it looks like I can't remember the exact percentages in that blue category that you were sort of skipping over, but it looks like mostly what you're picking up is fairly large effect mutations. And I wonder, even a 0.5% transmission advantage is huge evolutionarily.
yeah, yeah.
And I wonder, do you have thoughts about how many genes likely have a weak but evolutionarily significant transmission advantage? And are those genes somehow different from the ones your machine learning models predicted?
Y-
So, if they're the same as the genes of large effect, and your machine learning model's probably picking them up. But if they're somehow qualitatively different.
Yeah, and this is a great point, Jeff. Yeah, so we did leave out those. It's 45% to 48%. So those are, from my point of view, very weak. But of course, like you're saying, from an evolutionary point of view, they still would have an effect, and would be, I guess, selected against. And it's really hard to know. I mean, if you look, I mean, if I pulled the plot back up, what you'd see is that if you compare on either side of 50%, that there are way more points, there are way more alleles on the the less than 50% than the more than 50%, which sort of argues, I think, at some level that what we're seeing there is real. The problem is, since the effect is so low, I mean, you have to have more ears to actually, nail down that that's a real effect. However, I would say that if you look at that last plot, that the pollen non-specific genes really have a lot that are, at really near Mendelian effects. So I think I don't know what to say here, other than I think that our approach can't really answer your question. I don't think we can get enough data to say to what extent really small effects are present. I will say that the other reason that we sort of chose that the 45 and 48% uh, uh, sort of held out set is that Sebastian found that when he included those that were in that range, that the modeling didn't work as well. So that fits with your thought that there's maybe something qualitatively different, and that is the ones that are the ones that we're modeling on are qualitatively different in some way. Does that answer the question?
Yeah, I think that's along the lines of my thinking. Thanks.
Okay.
Ruben?
Hey, John, did you look at the impact of genes that are specifically expressed in silks?
Um, we haven't looked specifically. There are data in from Justin and from basically the surveys about expression in silks, and I don't remember any of those features. They would have been included in the modeling, but I don't remember any of them coming up as being predictive. Sebastian, do you remember if any silk features showed up? Why do you ask, Ruben?
Because I would expect that the things that are that are specifically expressed in silks may have an impact on pollen thickness, right?
Okay, so I would agree with you, and except I would say that those, since the silk is the sporophyte, you'd likely have to have a homozygote to have that effect. And we're only, in this case, we're only modeling the phenotypes that we get from pollen as a haploid. Does that make some
Yeah.
sense?
Yeah.
I think was I next on the list? Yeah. So I was actually I was kind of just thinking through, like, so maize pollen is definitely not as elaborated as like a bryophyte, but it is definitely a life cycle. And so I'm kind of like wondering, like, of the patterns of these genes, if you're gonna filter at any sort of step, like, are there certain like critical thresholds that seem like they might be like sort of cropping up? Like, is it like you're having issue with like sporopollen and wall production? Is it like you're not getting the cytoskeleton in order? Is it like the sort of redox chemistry? Because making that pollen tube is metabolically a pretty intensive process.
Yeah. So I guess I would say, yeah, I mean, a little bit, it feels like you're asking what were the genes that showed up with with phenotypes.
Yeah.
Yeah, and the types of genes that showed up are the ones that I think that you would expect based on many of them were. So there's some cell wall type genes, there's some cytoskeleton type genes. I don't remember if there's any redox type genes. Uh, and the other thing that's a little bit missing were genes that maybe would be involved in development of the pollen grain. I mean, since I mean, we focused on when choosing our targets, genes that were expressed fairly highly in the pollen grain itself. And so at that point, the pollen grain is really getting ready for pollen tube growth. And so I think that probably we're a little bit biased towards picking up genes there. If you come in 4 weeks and see Diana Ruggiero's talk, she's going to also discuss some of the mutations we have, and there's a really interesting mutation there called BAG1 that she'll talk about, which is looks like it's probably cytoskeleton something.
Awesome, thank you. I think Jeff, you had another one, right? It looks like Matt's was more of a comment than a question.
Yeah, I just missed what I think you had a comment about the effect of singleton genes.
Mm-hmm. Mm-hmm.
I just missed what that was.
Yeah, sorry. So, so we included in the modeling as part of the genome sort of derived features, categorized genes as either singletons. So there was no, the homolog's been lost, and paired. That is that both genes are still present. And in the final model, when you include those features, they end up in the model as you would expect, and that is that the singleton genes are sort of positive predictors of a fitness effect, and the paired is a negative predictor, so it's less likely to have an effect. In the final model, the paired feature drops out. It's not strong enough to sort of continue in the final model, but the singleton feature does, and it's a positive predictor. So, I think that, this was really, this is definitely one of my expectations, right? That if you have genes that are duplicates, that they're less likely to have an effect versus the singletons. So sorry, I did that rather quickly.
Let's see. Ellie, I think you might have the next question if you want to hop on.
Hi, thanks for an awesome talk. That was really cool. Pollen is amazing.
I'm on board with you there, Ellie. Thank you.
Yeah. Well, before I say the question I wrote in the comment, I do have some wild speculation, which is when I'm looking at some chloroplast and photosynthesis project stuff, it's always interesting to me how what's left over in the chloroplast genome is so important specifically for photosynthesis in the chloroplast. The evolutionary history of everything else getting purged kind of forces it into that shape. I wonder if in the evolutionary trajectory of the gametophyte becoming the less dominant stage over time, things that are not important for anything except just making the gametophyte also kind of get purged.
This sounds, yeah, yes, I agree. This sounds like a nice idea, and would sort of, again, I think go along with the specificity bit, right? I think one of the questions is whether is maize pollen just so extreme that it's really, it'd be interesting to know, does Arabidopsis pollen also show this same sort of thing where the specificity is such a strong predictor?
Yeah. So then, yeah, on that topic, something that's actually answerable. Yeah, I don't know what to do about that question or thought. It's just out there. But something that's maybe more specific is thinking about how there are these pollen genes that are like super, super highly expressed. Maybe it's because of the like dramatic length of the pollen tube in maize, but it could be because of this like yeah, maybe like just specificity of this tissue type. I know that Jonathan Gent had this project where he was looking to see different types of methylation patterns in like the 3 different cytosine methylation contexts in maize. And he had found that a lot of the really highly expressed pollen genes specifically had this kind of TE-like methylation pattern. And I was wondering if that might help you figure out some. I don't know if that was in your features you included from Maze GDB or not, but I thought maybe that could help answer some questions about like the TE insertions or what part of this is maybe more about the methylation status versus expression. But those are so correlated, maybe it's hard to disentangle them.
Yeah, thanks for that. There is in Maze GDB the data set that, they really that that resource is super incredible. They really sort of took the whole genome and sort of served it up on this platter of everything they could pull out of the genome. And one of them was the methylation data that they had at the time. And so I'm not sure whether it had the data that you're referring to, Jonathan's data. And so it didn't turn out that any of the methylation sort of pattern quantitative features showed up as predictive in the model. And I don't know, Jonathan and I have talked about that, and there are you're absolutely right. There's this set of genes that are super critical. There's a couple things about those genes. One is that a lot of them are actually in tandem duplications. And so it's unclear whether if you mutated them, they would have an effect because they're sort of multiple copies.
Yeah.
And the second thing is that's a pretty specific set of genes. And I think it's a relatively small set relative to the large sort of cross genome I think that we're looking at. None of the genes that were in our training set, none of our DSGFP insertions are in Jonathan's set of the methylation, the TE-like methylation genes.
Oh, I see. Okay. All right.
So
That makes sense.
we've talked about it and
Yeah.
we're sort of sad that they weren't.
That's really interesting though. You get like two views of what's going on with pollen. So
Yeah,
it could be complementary in a way.
I mean, I think it just points out that pollen is unusual and weird and interesting. But I'm biased.
No, it definitely is. Maybe I'm biased as well.
That's all right. That's all right.
All right. Well, I think that might wrap up all of the questions that are in the chat. If anybody has any like pressing last minute questions, otherwise, we might be good to adjourn. John, thank you so much for stepping up to present today.
Thanks for giving me the opportunity. It was really appreciate the chance and the feedback.
Yeah, definitely.
Let's talk, John.
Thank you.
Here, let me just kick this.
Bye.
Or here, I'm just trying to turn off the recording.